Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we're diving into the physiology of light detection, specifically the phototransduction pathway and the roles of photoreceptors, which are rods and cones. On the electromagnetic spectrum, there are a bunch of rays. You have X-rays, microwaves, gamma rays, all these rays, and you have the visible portion of light, the visible spectrum, which is a small portion, and it ranges from 400 nanometers to 700 nanometers. And this is what humans can perceive. The highest wavelength, like I said, is 700 nanometers, which is red, and the lowest is 400 nanometers, which is violet. And so light enters the pupil and it goes to the back of the retina, hitting the photoreceptors. Photoreceptors are specialized neurons in the retina that convert light into electrical signal. There are two types of photoreceptors, rods and cones. Inside rods and cones, there are thousands of optic discs that have proteins on them. In rods, there's the protein rhodopsin, and in cones, there's a similar protein called photopsin. When light hits the photopsin and rhodopsin proteins, then this triggers the phototransduction cascade. Now, rods or cones, they're used so you can see things at night and in the day. Now, in the day, when you have lots of light, you use cones, or primarily use cones, and this is so you can distinguish through the colors and you can have clear, sharp vision. You have about 6 million cones, and they have a fast recovery time, and they detect color and fine detail, but they do require bright light. At night, when you have less light, you'll use rods. Rods are highly sensitive to light, so they can detect things, but in grayscale. So imagine you're watching a black and white movie. You'll still have some cones, you'll still be able to detect some color, but not in the way you would be able to when there's a lot of light. There are 120 million rods where there are only 6 million cones, and rods are 1,000 times more sensitive to light than cones are. As we talked before, rods are packed in the periphery. I guess they're dispersed in the periphery, and cones, they're packed in the fovea. There are 3 types of cones, red, green, and blue. 60% are red, 30% are green, and 10% are blue. And so cones are primarily found in the fovea. The fovea is the center of the macula, and it only has cones in it. There are some rods in the macula, but there are none in the fovea, and the rest of the rods are in the periphery. Now you have 3 types of vision: photopic vision, mesopic vision, and scotopic vision. Photopic vision occurs at high levels of light. Mesopic vision occurs at dawn or dusk and involves both rods and cones, and scotopic vision occurs at very low levels of light. You can remember this by thinking photopic sounds like photopsins. Photopsins are in cones, and so they occur at very high light levels. Mesopic is like the middle, so you have both rods and cones, and scotopic is not photopic or mesopic, so it must be rods. Now, let's talk about the phototransduction cascade. The phototransduction cascade has the same inherent process in both rods and cones, but for the reasons I discussed earlier about them having a different recovery time and having different light sensitivity, they occur at different times. So I'll tell you about the phototransduction cascade in rods, but it's essentially the same in cones, and all the proteins are essentially the same. So rods are normally turned on, but light hits them and turns them off. The process of turning this off, the rods off, is called the phototransduction cascade. Now, we talked about light hitting the rhodopsin and photopsin, and these proteins have a molecule called retinol. Retinol is derived from vitamin A, and it's normally found as 11-cis-retinol. When light hits the photoreceptor, it changes 11-cis-retinol into 11-trans-retinol. Retinol changing shape causes rhodopsin to change shape. This activates the rhodopsin, which then activates a protein called transducin. Transducin is a G protein, and recall that G proteins have alpha, beta, and gamma subunits. The alpha subunit of transducin then activates phosphodiesterase, or known as PDE, and this breaks down cyclic GMP into GMP. In darkness, high levels of cGMP, or cyclic GMP, keep sodium channels open, allowing sodium to flow in. This makes the cell stay depolarized and continuously releases the neurotransmitter. But with less cGMP, sodium channels close and it turns it off. The cell then hyperpolarizes, reducing neurotransmitter release. And you get less cGMP when transducin activates phosphodiesterase to break it down. You get transducin when rhodopsin is activated, and rhodopsin gets activated when retinol changes shape, and retinol changes shape when light hits the photoreceptor. And so to recap, the last thing that happens is that when less cGMP, your sodium channels close, you turn it off, the cell hyperpolarizes, and you reduce neurotransmitter release. This change in neurotransmitter release signals to bipolar cells that light has been detected. Now there are two variants of bipolar cells. You have on-center and off-center. On-center are active bipolar cells and off-center are inactive. So when on-center bipolar cells are turned on, this activates on-center ganglion cells, which sends the signal to the brain. I want to make a quick distinction between color and brightness before the end of the lesson today. So we tell color by the ratio of cones turned on, and brightness by the amount of cones turned on. So for example, if I have 100 red and 70 green cones that turn on, the color would be yellow. Now, even if I have 10 red and 7 green cones turned on, the color would still be yellow as the ratio stays the same, but the brightness would decrease with less cones turned on. Now to summarize, photoreceptors detect light and convert it into an electrical signal through the Phototransduction Cascade. Rods must be off to send the signal to bipolar cells, and they're considered off because sodium channels close. Rods are for sensitivity and darkness, and cones are for detail and color, and there are 20 times more rods than cones. In the next episode, we'll switch senses and talk about the auditory system.